There's always something new to learn or discover, and biology always finds a way.
Yes.
That's the greatest thing about being a biological engineer, is that biology evolves. We wouldn't be sitting here if it hadn't evolved and kept changing to meet challenges.
Erum, what is going on?
Oh, Karl, I don't know. I mean, it's seeming like the simulation is revealing itself.
So the simulation is bleeding into the Grow Everything podcast?
I don't know. The algorithm's been feeding me a lot of information about simulation theory and the prompt theory. I don't know if you heard of that, where we're basically living out prompts from AI, and we are the digital beings, and biology is technology. I feel like I've just been being fed this information, and I'm like, what is going on?
All right, well, I don't get those, though I do get a lot of AI videos on YouTube that are more about setting up LLMs and how to create prompts, and actually reviewed some of the prompt that this company that I think is in the AI space developed. And they talked about how they use different AI models to develop these prompts, and their prompts are super crazy long. I'll have to find it and share with you and share in the notes, but I was really impressed by the work that they've done. But Grow Everything is not necessarily about prompts, though it could be about the simulation breaking through.
Yeah, no, it's just every day we're living out a storyline, and it's really interesting. And we're exploring how biology is the platform in which we experience life. And it's great to work with it. I mean, that's what we're doing at Grow Everything, looking at how biotechnology is manifesting in this simulation. But we've had a lot going on. I mean, I'm glad everyone, you, the listeners, tuning into this. And we have some exciting stories. I mean, first, let's talk about what have you done recently, Karl?
So over the weekend, I went to the Brooklyn Waterfront Artists Coalition opening for their Fungi art exhibit. My wife, Kristen, who is a public school art teacher, also is a painter, and she has some encaustic works. Encaustic is painting with and on wax. I actually posted a reel on my personal Instagram that shows a quick walkthrough through part of the exhibit. But it's just great to see these tributes to mycelium and mushrooms and lots of mycelium art. GenSpace actually teaches mycelium art making, how to make art with mushrooms. So it was really good to see the artwork that was being displayed in tribute to the almighty mushroom. So that was one of the things I did. I thought it was really pretty stupendous. So last week we mentioned Joe Rogan's interview with Ben Lamb of Colossal, and he was talking about using synthetic biology to bring back the woolly mammoth. And this is something that they've been working on for years. They recently introduced these dire wolves. And if you pay attention to the news, you see that they've already engineered these woolly mice. All of this stuff gets talked about on that podcast. And it highlights very publicly the power of synthetic biology and what it's able to do when you're able to engineer biology. On this podcast, we've had several people come on and talk about using engineered biology or synthetic biology for conservation purposes. Particularly, we had Ben Novak of Revive and Restore, and there was recently a paper from Stanford talking about using synthetic biology to address sustainability challenges. I thought it was fascinating, and I thought that people who listen to this podcast might be interested in that. Very easy to read article. We'll link to it in the show notes.
Nice. Yeah. I mean, there's a lot of episodes that cover how we can create different products and how they can be not only more sustainable, which in our discipline of leveraging biology in a smart way, we see sustainability as table stakes. So we're really looking at functionality, but it's great to share more information about how sustainability is one of the core principles of leveraging biology.
Yeah, it's biology is the ultimate manufacturing platform. Not too long ago, we had Alexander Titus, who was on the National Security Commission on Emerging Biotech on the podcast. And today we're having Angela Belcher, who's an MIT professor who was also on the National Security Commission on Emerging Biotechnology. She's a professor, and what she's known for, at least in our world, is innovating material science by using genetically engineered viruses to build advanced technologies. This idea of using a virus to build things is amazing. And she's done applications in energy, meaning batteries and solar cells, electronics. She's worked on environmental solutions, and she's very passionate about early cancer detection. So this idea, if you can imagine that there are natural processes. Biology is out there building seashells. That's how she got fascinated with this idea. How did we end up interviewing Angela Erum?
So we got connected to Angela because we went to a biosecurity meetup. Remember that we collaborated with Aclid Bio, so our friend Kevin over there, and Andrew Hessel. There was an amazing person. Her name is Allison Check. She works at the National Security Commission on Emerging Biotechnology and incredibly smart. So you should listen to the podcast. She came to us and was telling us about the reports that they were putting out and introduced us to Angela because of Angela's background. And you guys will see that we could probably do 3 different podcasts with her since she is looking at biology in so many different ways. It's going to be great to hear. With that, let's let Angela Belcher take it away.
All right, Dr. Angela Belcher, MIT professor, UC Santa Barbara alum, and distinguished so many other things. We are so proud to have you on the Grow Everything podcast. Welcome to the show.
Thank you, Karl. It's amazing to be here and nice to meet both of you. Nice to meet you, Erum.
Meet you too.
I'm going to kick it off. You've worked on so many things over the course of your career. You worked on energy, you worked on health, you've worked on a lot of other things before that. How did you get started and what got you interested in biological engineering?
So thank you for the question. And it's a long answer, but I have been in love with how biology makes materials for a long time, really since middle school. I've been fascinated about things like abalone shells. I did my PhD. You mentioned I went to UC Santa Barbara, right there on the ocean. I could see the whales and dolphins that go by from my bench as a graduate student. And I was so fortunate to have a PhD project, which was how do abalones make shells? And I think, was that a marine science project? But no, it was a material science, chemistry, molecular biology project. And a lot of what I do now and my love for the interaction between biological organisms and biological molecules and inorganic materials comes from my time thinking about how abalones grow shells. If you've never seen an abalone shell, I have one here, but I can also describe it. Abalones are marine gastropods. They're marine snails that live in the ocean, and they make really exquisite shells. And these shells are made out of the elements that are in the ocean. So they're made out of calcium and carbonate. And the organisms in the ocean, including abalones, evolve the ability to basically retrain their proteins to grab calcium and carbonate out of the environment and build really exquisite structures on the nanoscale. So that's cool and interesting all on its own. But when it does it, it uses non-toxic materials, non-toxic precursors, doesn't add toxic materials back into the environment, and makes them in a way that makes them 3,000 times tougher than their geologic counterpart. I just think that is amazing. If you think about how our bones are made and how our teeth are made or how trees and bamboo are made, it's all controlled at the genetic level. They have such exquisite properties. So I've dedicated my career to encouraging biology to do even more. What if it could have done better? What if it had the whole periodic table to work with? What could we build with biology?
Amazing.
It is so amazing.
You are the perfect person to be on this podcast. Yeah. We're talking about growing everything. You're looking at things from the atomic level, at the biological level, and then having to bring it in society and have it function and to be able to think that way. We love people that to come on here and think that way. You have this amazing career and you've worked on several projects. And I'm curious, what is a project you never expected to work on and how did you end up working on it?
Okay, do I have to pick one project for this?
No.
3.
Okay, 3. So I really started out as a basic scientist. I'm a chemist like you.
Woo!
And I really worked on trying to unravel the principles of how abalone grow shells, which was amazing to get to work on, like isolate the proteins, study the crystal structures that they make and how they shape it, how they form it. So it was really a basic science project, which is great for a PhD project. But at the same time, I remember the day that I was actually standing on top of my desk as a graduate student looking at my periodic table and thinking, wow, biology's done such an amazing job with calcium carbonate and calcium phosphate. What about the whole periodic table? If it worked with calcium carbonate and calcium phosphate and iron oxide, why can't it work with semiconductors? Why can't it work with magnetic materials? And so I jumped off from there to trying to combine the idea of selection and directed evolution, but for semiconductor materials and for solar cells and for batteries. And here's an example of a battery that we made, which was a big surprise. This is a genetically coded battery. I'm holding it here. It looks just like a coin cell battery, but it's powering a little LED. The anodes and the cathode materials from this are coded at the genetic level with bacteriophage, which are viruses that only infect bacteria. Instead of having a bacteriophage make a shell, we said, oh, why don't you make an anode? Or why don't you make a cathode material? Oh, and then why don't you wire yourself to the current collector and make a fully functional battery? People called that idea insane. I've been called insane quite a bit. But for us, because we're in this life and in this sphere of thinking that biology makes things amazing, it wasn't that big of a jumping off point. But did I ever think I'd be working on batteries? I did not. But then once you start working on batteries and you start looking at where things are related on the periodic table, then the sky's the limit or the ocean's the limit. One of the things I'm most proud that I'm working on now that I never thought I would be working on is I have a 10-year goal of working with amazing collaborators. So I do nothing myself. Everything is with my students, my postdocs, my collaborators. But I have a 10-year goal of developing detection for ovarian cancer. So I never thought I would be working on that, but that's something that I'm obsessed with. And I will end my career working on. Hopefully I'll end that before the end of my career and then move on to something else. But that's something I plan to be on in the next 10 years.
That's amazing. I just have one question going back to abalone. To me, one of the most amazing things about those shells is the inside is multicolored. So is that a function of the way the proteins are laid out? Are they using chromophores? Is it the way that they reflect the light? What is it that causes the color, and how do the abalone actually build that color function into there?
Yes, yes, and yes. And so
Okay,
abalones are quite brilliant. So most of it is actually structural color. And if you have an abalone at home, which I hope some people do, you can take the abalone and you can put it under a light and start to rotate it. And those beautiful colors that come out of it, I have a red abalone here that comes from the coast of Santa Barbara. I have New Zealand abalone as well, and they're slightly different colors. When you rotate it, you'll see some of the colors, the blues and the greens and reds rotate through. Now, that's structural color, meaning it doesn't come necessarily from a pigment. It comes from the thickness of the calcium carbonate tablets. If you think about pearls and if you think about shells, they're made out of two different crystal structures of calcium carbonate. The outside, the red abalone or the black abalone, is calcite. So that's the thermodynamically favored structure of calcium carbonate. The inside, that mother of pearl-like structure, is made out of aragonite. That's a metastable crystal structure. I do have crystals here. Here's what aragonite looks like. It looks like a pillar, a column. This is what it looks like geologically. But what it looks like biologically, when it's made by an organism, it looks like a stack of coins that are laterally offset to form a brick wall. That's part of its secret. That's what makes it so tough, is this stack of coin-like structure. And between each of those coins, if you think about a quarter or some kind of coin, all around that coin are proteins. And those proteins serve many functions. One of the things that proteins serve is they act as nucleation sites to actually grow calcium carbonate in the right crystal structure. But they also have complete control over the thickness of the tablet. And the thickness of a tablet are on the order of wavelength of visible light. So it's an optical effect. The organism makes them very uniform, the right thickness. And so the colors that are coming from most of it is the interaction with light. And that's why the colors move around. Now, when you go home, you're going to go home and you're going to say, well, Angela said it was all based on optical properties, but here I see this color doesn't move. Well, some of them actually are pigments. And those pigments are cross-linked, also cross-linked amino acids, and they come from what they're eating. So what kind of kelp they're eating, things like that.
So amazing.
That is so fascinating. I want to enroll at MIT somehow and be one of your students, because I could listen to you talk about science forever. But you do so much more. So in addition to being a professor at MIT, you serve on several national academies and have started multiple companies. So for the last 2 years, you served as commissioner on the National Security Commission on Emerging Biotechnology alongside Alexander Titus, who we are big fans of. We had him on our podcast several episodes ago. So for our listeners, we recommend you guys listening to that and get Titus's perspective. He likes to be called Titus. But for our listeners who missed that episode, can you refresh us on the commission, its mission, and what this new report's all about? Because that's been big in our industry, this report that you guys just published.
Okay, yeah, thank you. And I love working with Titus. He's a delight to work with. He's so brilliant. He's a great speaker. He's such a forefront thinker for the Department of Defense needs and did a lot for bringing biotechnology into basic science and translation for U.S. needs and around the Department of Defense. And so it's been such a pleasure to work with him. So in general, commissions are brought about by Congress when Congress wants to do a deep dive on a particular topic to understand what the state is of a particular topic in the country and in the world and what the bottlenecks are. And then the job of the commission and the commissioners is to put that into a report and to bring forward recommendations for the government on how to best situate ourselves as a country and policies to make an impact in a particular field. So in this case, it's the National Security Commission on Emerging Biotechnologies. That's a big topic. Emerging biotechnology is a big topic. And our commission is special in that it's bipartisan and bicameral. We have both Congress people and senators on it and both parties. And so that's integrated into our commission. We have 11 commissioners, including like Titus and I, who are academics, but also people from industry and from national security and the intelligence. So it's an industry. It's been an amazing group of people. So amazing group of people that came together for the first 2 years to think about emerging biotechnologies, the impact on national security, and the impact on the nation as a whole.
What was the origin of your participation? How did you get involved?
So as a professor, I wear a lot of different hats. Probably my favorite is teaching. I'm an undergraduate teacher and mentor, and then I run a research group. But in addition, as you get older and more senior in a field, you get asked for your opinion and you get asked to make presentations a lot across the government. And I've been doing this for a couple of decades. So I have the opportunity quite frequently to speak to members of Congress and members of the Senate and their staff. And it was through one of those conversations and presentations that I was asked to be a part of this commission. And I have to tell you, I have lots of different feelings about this. One is that you first think, okay, this is pretty important and it's pretty interesting and pretty cool to be a part of this. I think we met with 1,800 people in 30 countries across lots of different areas of biotechnology and beyond biotechnology. We got to understand where things are in many different fields, anywhere from basic science to the government to translation to policy to agencies, what needs were being met, which needs weren't being met, the aspirations are where we need to be and what the bottlenecks are to getting to that point. In that way, it was just amazing. And I learned so much. It was like getting a master's degree or a PhD or something in this particular area. I didn't estimate how much time it would take. It took a lot of time, actually, but it was well worth it. And it's the service to our nation, service to the country. And I feel very passionate, it's probably easy to tell, about biotechnology and the future of biotechnology and emerging technologies in general. So that's how it came about. But it's the most work of any committee I've ever been on.
Wow, Angela,
That's amazing.
Thank you for your service. I can imagine all the vectors that you're mentioning to be able to think of the bottlenecks, opportunities. We talk a lot about different emerging biotechnologies here on the podcast and have the people building it on. And a lot of times there's been the perspective of entrepreneurs, but again, we had Titus on and people from the other areas. But of all of your conversations, this report came out. What we want to know is, what recommendations are you most excited about?
So, okay, again, you asked really hard questions.
We're really good at that.
Okay, well, that's a good skill. Let's see how good I am at answering that. I think we had almost 40 recommendations from this. And one thing that's interesting to point out is that the recommendations that we put forth or in the report were unanimous recommendations. That means you have 11 people from very different backgrounds who had to come to consensus on, yes, this is important enough, and we agree with what it is that it makes it to the report. So this wasn't something we just thought about. This is something we thought about a lot. We debated, we refined, and put forth what we thought was going to be most important. And I'm not a policy person. I was forced to learn some about policy, and it was definitely interesting. But my passion really is around research, translation of basic research into products. And I'm an engineer, so I don't make anything that's not useful. And then around education. And people have asked me this before, and I always feel like it's like, which of your children do you love the most? Or cat, or whatever. But some of the ones that really come to mind are one around, we call it the Parks Initiative. And what I really love about this recommendation is it brings together so many different aspects, but it focuses on public lands. For example, it could be the national parks, but it's beyond the national parks. And what it revolves around is basically sequencing the amazing diversity of our public lands and using that in a way that serves the nation. First of all, I love national parks and spent so much time in graduate school actually doing field research in a national park, and then growing up camping, hiking in different national parks and state parks. But you both also know that there's incredible organisms, whether they're plants or whether they're microorganisms, that live in really interesting environments. And we all know that polymerase chain reaction, which PCR and the enzymes around that came from our national park. It's amazing. They came as extremophiles from our national park. So there must be many, many more that are available. Another aspect that I love around this is that these parks belong to us. They belong to all of us. It's national land. So as we are going through and sequencing and preserving and learning about the different organisms and species, the data that comes out of that gives us the opportunity to start standardizing data and thinking about how do we take this data and standardize it and allow it to be used by people in industry, people in academia like me, that we all have a right to use that data. And we all have a right to think about designing the next important material that might come from a new plant that we didn't know very much about, or an organism that we didn't know about, or enzymes that could be used in manufacturing. So that's one aspect of it. The other aspect, this is an idea that I have, and Titus is on board with this as well. We actually sat next to each other a lot in these commission meetings, and he's both fun and distracting to sit next to. A lot of us are really interested in thinking about how to inspire the next generation. An idea that I've been dreaming about around this was, wow, what if every elementary school in the country could adopt a few organisms from the national park, and they're in charge of the DNA sequence around that organism? And what if they're in grade 3, room A, they're in charge of these enzymes? And so they have to think about, how do you go from a DNA sequence to a protein sequence? And what if you were doing that in 2nd grade, 3rd grade, 4th grade, something like that? And thinking about, okay, you have to learn about how that works and what all the processes in and the basic science around that. But if you're working on a plant organism, you get to learn about photosynthesis and you get to learn about ecology. And I think it could be really cool if a person or a child could own the ability to understand and publish, if you will, about their organism. So I think it goes into bioliteracy, which is something I'm very, very interested in. And so I think it hits a lot of the recommendations because it's about data standardization and data storage. It's about how do you access data. It could be around scale. We can talk about that in a minute. But it's around bioliteracy, because I even think about kids getting little badges and stickers around, well, I've worked with sequences from these different kingdoms and things like that. So I think it would be a lot of fun.
That sounds like a lot of fun, and I want to participate. I was in Girl Scouts. It was about starting a fire, setting up a tent, but to be able to do microbiology and adopt a microbe. I could even see the enamel pins. It's so cool.
Yeah, I see that too. I see the little badges. You're like, oh yeah, you have your E. coli badge. I have my
Pseudomonas or whatever.
Yeah. I think that would be so cool. And so
Love it.
I'm passionate about teaching science, and we have workforce development in this report, and we have engaging grades 9 through 12 in biotechnology for all. And that's around, how do you think about a curriculum that would be accessible across the country, and how could you incentivize teachers to think about this and participate in the latest hands-on ways of teaching. Also, the bioliteracy and the workforce development is important in community colleges and technical vocational schools, which I think is really important, because when I start thinking about, and I'd like to hear y'all's perspective on this, because when you start thinking about when we're going to be making everything with biology, or a lot of things with biology, it's going to require a lot of different types of skills. And some of these skills will require a PhD in molecular biology. Some will require a 2-year certificate in manufacturing. Some of them will be analytical chemists too,
Yes.
to be able to study what's happening and changing during the process, and chemical engineers. And it's going to take so many different people with so many kinds of backgrounds. So how do we prepare for that path? One of the things I'm really interested in as well is workforce retraining. Can we take people that have been in different fields and give them the opportunity to work in biomanufacturing? And I don't believe that requires a 4-year degree or a PhD, but thinking about what those skills are, and those skills are transferable, and you could live in Massachusetts like I do and then work and then transfer them to Ventura area or California and be able to carry that skill set with you. So I think that workforce development is a huge part of what we're interested in. But my passion at the end of the day, besides ovarian cancer, my passion at the end of the day is it is so much fun to teach kindergartners about protein synthesis.
Wow.
I've done that. I've given them amino acids, like, okay, you're alanine. Give them a little placard with their amino acid on it. And then we have a ribosome, and you go through the ribosome and you build your polypeptide chain. I think that kids love that. It gives them an insight into what's inside of them, what makes them who they are. I think more of that would only be a blast.
Yeah, you're inspired me because my son's going to preschool. So he's getting to that age, and I want a kit that has the building blocks, has amino acids in it, so he can start learning because he absorbs information. A little sidebar I'd like to tell. I'm a proud mom that Karl, he is a very smart kid. I'm not just saying
He is. I know he is.
I'm saying it because, he knows every single country in the world. He knows their capitals. Wow. He knows their shapes. Wow. He can look at just a bunch of dots and he'd be like, that's in Marshall Islands. And I'm like, how the heck? So he's pretty good, but I feel like he could do this with chemistry and biology. So you planted the seed in my mind, and I feel like now it's my duty as a parent, as a woman of science and chemistry, to bring that into his life now. So
I think that's super, super fun. And we can brainstorm around that. I teach with dodgeballs. I call it dodgeball science, because dodgeballs, when I was a kid, they were all like red, but now they're all these beautiful colors. They're purple and they're green and red. And you can almost teach anything with a dodgeball, because dodgeballs are photons, they're electrons, they're atoms, they're amino acids. You can build polypeptide chains with them. You can build a crystal structure on the floor with them. Is it amorphous or is it crystalline? We play semiconductor dodgeball. The ball is a photon, and you try to excite an atom.
Yes.
Because as you know, little kids, a lot of them like to be active, and electrons are active.
Yes.
Photons are active.
Yes.
Yeah.
I love it.
Yeah. My wife is a public school art teacher and does K through 5. And so I get to hear the stories of the things that they react to, how excited they are to learn different things. And it's another way of teaching the curriculum is through art. But I'm so fascinated by this Sequencing Public Lands initiative, because what it reminds me of is at the turn of the last century, so 1890s, as Theodore Roosevelt was growing up, there were these initiatives to really understand the animals of the American continent. And I think the Smithsonian was started by people just sending in samples to Washington, D.C., to what would become the Smithsonian. So there was this call to action, which was taken up by young boys. I think Theodore Roosevelt was given his first shotgun when he was 8 years old. And so these kids are all shooting birds and sending them to D.C. And he had this huge collection of birds and animals in his house somewhere in Manhattan, and that became the basis of the Natural History Museum. So just imagining the ability to take this biodiversity project, collect out this data, the public good that would be, is just so amazing.
Hi, a quick shout out to our sponsor, Messaging Lab, the force multiplier for biotech. Your biotech company is making the world a better place. You know that, we know that. But does the world? There's a big reason why some biotech companies attract investors, sign up customers, and get attention. That reason is strategic communications. At Messaging Lab, we translate complex science and economics into compelling narratives. And we have done that for the most successful biotech companies across healthcare, agriculture, personal care and beauty, materials, and the list goes on. We're here to make sure your ideas not only get heard, but resonate with your audience. So if it's time to amplify your company's voice, visit messaginglab.com to explore how we can elevate your story and grow your business.
Yeah, thank you. I don't really want to shoot any of the animals, but
or the microbes.
I didn't say that.
yeah, I know. I get that. And
Yeah.
I think that's a great analogy. And honestly, the Smithsonian Museum is one of my favorite places on earth. So it's great to see that. Okay, so the other one is around grand challenges. That's another one I worked quite a bit on because the idea is what can we do to capture the imagination of people at all levels? So it'd be, academia and industry and the government and people in general. So two different grand challenges that we put forward, which is one that I think that y'all will both love, is how do you make engineering of biology more predictable? And that, again, the idea is we spent decades doing amazing work around unraveling the basic biology and biochemistry and around how biology does things. Talked a little bit about the abalone shells and many different things like that. But how can we do that in a really predictable way so that we can make products, which people already do, but make products in a predictable way that I have a blueprint for it and I can make it here in my lab in Massachusetts, and then I can give Karl those blueprints and he can make the exact same molecule because it's been done in a very predictable way and a way that is scalable. And I think the thing that I like about this particular one, it's always hard to say, like, how do you pick two grand challenges? But what I like about this one is that it incorporates so much science, engineering, and technology. So it's not just for people who are biological engineers like me or even life scientists, because you can think about how it touches so many different areas of engineering and science. You can think about physics and manufacturing. And you can think about electrical engineering and computer science and AI. It touches so many different fields and so many different topics because so many people have things to contribute to this if we're really going to get to the way of making things very predictable. This has been done in electrical engineering and computer science and many different fields. But I think that there's still a long way to go to make it in a way that will really revolutionize how we make bioproducts, whether they're molecules or therapeutics or building supplies.
So that was one of the grand research challenges. What's the other one?
The other one is around scaling biomanufacturing.
Yes.
There's multiple components around that, but there's still actually science that needs to be understood and better facilitated in order to have things scaled in a really predictable way. So there's the science around that. We also have very important components in this report around increasing the capacity and availability and accessibility to scale biological products, not just in one or two regions around the country, but how do we do that in a distributed way around the country to where a small startup company, a medium-sized company can come in and test to scale their product and then scale their product. And how can we do it in a way that onshores brings the whole supply chain around it focused in the United States? Because that would definitely have national security implications. It would definitely have the ability to increase jobs and new kinds of jobs locally to where people all around the country could be involved in manufacturing, but you don't have to move to one of the two coasts or something. This grand challenge goes into that in the fact that it focuses on what do you think about the science of feedstocks and chassis, what organisms are going to be used, what are the critical inputs, what kind of technology needs to be done in order to measure things in real time while you're scaling and make decisions along the way.
Wow. I'm just going to say, I know Karl's favorite topic is distributed manufacturing. So go, Karl. I'll leave it to you.
No, I mean, Angela, you said it. The idea is that you don't have to go to one of the coasts to be able to manufacture. And we know we've got this incredible source of feedstock in the middle of the country. And there's different feedstocks all over the country. So it would make sense that manufacturing capacity does end up being distributed. And we've had a couple of episodes where we talked to people where we're like, what would it look like if your facility was next to the factory that is using the ingredients that you're producing?
That's exactly what we're talking about. That really captures our country is a big country. The kinds of feedstocks that go around this country are localized in particular areas. And how can we capitalize on that to basically make manufacturers stronger in a particular location? It's exactly what we're interested in doing. You said it better than I. Maybe I can use your words next time, but it's an amazing opportunity. Think how much fun that would be. You have an idea. I'm going to try it, but 10-liter scale in my lab and it works. Okay, now I want to take it up to 100 liters or 200 liters. If I could just get on a list, or if I could just go 20 miles from here or less and be able to test it out and have it be a go, no go. Well, that was a bad idea. It won't work. Or that's a fantastic idea. How do I make it to the 2000 level? Or how do I make it integrated into devices or products that can be used today? If we could do that all within the United States and it would be available to everyone, I think it would be game changing.
Yeah, absolutely. And I mean, I just am thinking in my mind, mapping like, oh, we've got feedlots in the Midwest. What is all the waste that's being generated there that could be used as feedstock for organisms? You've got this incredible richness of forests and different kinds of forests all over the Southeast. And then you've got the corn belt, which has all the corn. So you really have these unique feedstocks all over the country. And like you say, if you're able to go from the lab where you're doing milliliters to liters and knowing that you just maybe flip a switch and you've got the feedstock available to go to 1,000 liters at a time, I agree with you. It's 100% a game changer, I think. And it enables so much because then you could say, well, we know it works with this feedstock. If we could modify the organism, then you can just ship it across the country and you can use that feedstock as your input.
Yeah, I think it'd be amazing. I want to do it now.
Yeah. Yeah, exactly. I mean, that's how we feel. We're like, oh, we have our consulting firm, Messaging Lab, and we have this podcast. We're like, now we just want to get our hands and go in the lab and do the thing. So maybe we'll expand our offerings here at Messaging Lab.
I do have a little material here that I made. It just looks like carbon, but this is a material that we made that's a very high surface area carbon.
Mm-hmm.
I'm not sure which. I have one that's higher than this, but it's made by biology. We either use it making bacteria or we use it making bacteriophages. We can change the surface area anywhere from about 1,500 meters squared per gram to now 7,000 meters squared per gram.
Wow.
Really high surface area. And we made it originally for the Army to neutralize really bad agents like chemical warfare agents, but we resurfaced it. We reprogrammed it to work on many different things like breaking down PFAS and forever chemicals and gold mining and things like that. But the reason that made me think about it is that we feed them waste. So my students go to the brewery down the street and they get the waste from there or from the apple cider waste, and they feed it to our organisms that use it and make high surface area carbon that we're using to clean up either toxic waste or to mine important minerals.
What does that exactly mean? Just so our listeners understand, what does it mean to have a high surface area carbon? I almost see a futuristic, like you showed a vial that had a black powder in it. You take that vial and it's so small, but then when you spread it out, it ends up covering your house.
So here's a big bottle we have actually here as well.
That's like $1 billion right there.
Yeah. So the surface area is anywhere from about a hockey rink of a surface area per gram to a football field.
That's just crazy.
Yeah, so it's very, very high surface area materials that are made by biology, because biology makes it, you can functionalize it and do all different kinds of things. You can repurpose it for what we did with the Army, and they scaled it to 2,000 liter reactors there. But then you can go and clean up waste or trying to recover gallium and indium with it.
That's amazing. Again, biology never ceases to amaze me. Even when we've solved it, if it is a solution, the applications are just infinite.
It's never boring.
Yeah.
Yes.
I've never woke up and been bored in 25 years. I mean, because there's always something new to learn or discover. And biology always finds a way.
Yes.
That's the greatest thing about being a biological engineer, is that biology evolves. We wouldn't be sitting here if it hadn't evolved and kept changing to meet challenges. That's why I feel like it's endless, because we will be able to figure out how to evolve, how to help organisms help us solve problems.
It's so refreshing to hear these recommendations from you. We looked over the report, and reading is one thing, but to hear you frame it from your perspective, we learned about the bioliteracy recommendation, the sequencing public lands, the grand challenges recommendations, which I love. I think healthy competition. We live in the society, it's a capitalistic society, but yes, competition does spark the imagination. But other than that, again, you have nearly 50 policy recommendations for biotech, and some of these ideas already have been introduced as bills in Congress. But what's next for the commission? You already did all this work. What's the next thing?
So I thought it was going to be a vacation, but that doesn't seem to be working out. So the way that this commission was put together, which is very different than I think most commissions and is different than all committees that I've been on, is that we had policy experts with us on the committee. So when we came up with ideas and framed them, we framed them in a way that they could go directly into policy recommendations that could be picked up by members of Congress. And so at this point, what we're doing is kind of a roadshow in that we're going around and we're talking about what we found, what our recommendations are. And then we're hearing from people in terms of how they feel about those recommendations and are there additional considerations we need for the future. And so in a way, this is a little less work than it was before because it really is going to talk about the wonderful things about biology, which is the fun part, and trying to implement these recommendations so we can do exactly what we talked about today. So we can be one of the major biomanufacturing countries in the world where we can have people at all levels and all ages understand biology, whether it's understanding biology for themselves, for their own lives, for how their bodies work and such, or understand that it's the future to designing things in a new way to make an impact on people's lives. As far as I know, that's what's next.
Wow.
And what about for you personally? What's next for you, Angela?
So I'm still a professor. I just got my grades in yesterday for the term, which is
Good
great.
for you. It's a lot of work.
It was a lot. I was teaching seniors this term, and I haven't taught seniors before. I usually teach sophomores, and it was so rewarding to teach seniors because, wow, they learned so much in that time. And not to brag, but our undergraduates at MIT are quite exceptional. I feel so good about where we are because I'm thinking, okay, these are the future leaders. We're okay. They're smart, they're creative, they're passionate. Grades are in, yay. So I'm an inventor. I just love inventing. That's my favorite thing to do besides teaching. And right now I'm obsessed with clean mining. I'm working on that right now. We have a really fun program that sounds boring, but I think y'all won't think it's so boring, which is how to separate selenium from rhenium. And that's pretty specific, but that's from the mining industry. And rhenium is important as a material for engines, actually. And rhenium is expensive. It's a couple $1,000s a kilogram. And selenium is really inexpensive. But the company that I'm working with says it's hard to separate these two materials. And I'm thinking, gosh, biology should be able to separate those because we separate things that are much closer chemically than that, or otherwise we wouldn't be around. So we've been doing that, and it's so much fun. I love it. And then we're working on the rare earths and things like that. And then every morning when I wake up, I think, how much closer are we to solving ovarian cancer? How can we help women prevent ovarian cancer? And if they do, how can we help them intercept it early on? That's what keeps me going every single day, is thinking about how do you get closer to that? That's something that we can take off the table, is not being worried about.
Yeah, I will say that at Messaging Lab, we focus on biotech. This podcast is about biotech. We're over 130 episodes, and you just re-inspired me in thinking about biology and supporting these companies and organizations that we support. So the commission's very lucky to have you. MIT's very lucky to have you. Thank you so much for taking the time to speak with us today.
Yeah, thank you, Angela.
Well, thank you so much. And you were so much fun to talk with. I hope we get to talk some more. And I'd love to brainstorm around preschool. I did a bunch of preschool STEM education, and I'd be happy to brainstorm about that. But thank you for all you're doing for the industry in our field. We need to inspire people and focus on the great work that's being done across our country.
Absolutely. Thank you so much, Angela. We really appreciate it.
Thank you.
So, Karl, what'd you think of that episode?
I think I said it at the beginning of the episode. I've known about Angela for a really long time, so I've fanboyed her, following her and her career and the work that she's done. I didn't realize she had done so much work or was so passionate about ovarian cancer, but I was very excited to hear her talk about her passion for education, because we know how important education is when it comes to getting more people interested in what is possible with biology, which is part of the reason why we do this podcast. So amazing interview, amazing person. And as you said at the beginning, we could have 3 interviews with her. What was your take, Erum?
I love the education piece too, especially getting kids interested in learning biology at a very young age. She was talking about preschool age. That's my
Yeah,
son.
crazy.
And I think he could do it because, again, he has really good memory. He's curious. He likes frameworks. He loves things about the solar system. So he did a deep dive into that. He loves country. So he deep dived into that. I think he could deep dive into biology, especially if it's that we talk about the biology stack. What's this? A simple stack. That's the central dogma. And I think through visualizations and training in interesting ways, she was talking about using dodgeballs. I was like, what?
Yeah. She said dodgeballs. That was so funny.
I was like,
But
cool.
we've had a couple people talk about education. I don't know if we've had anybody talk about exposure to children at that young of an age,
Yeah.
but I'm pretty sure either Natalie Kudell or Christina Agapakis has said, we're all scientists.
Mm-hmm.
And the sooner you get exposed to the idea that we're all scientists, it demystifies the idea of being a scientist, and also, I guess, makes it more interesting or could promote you to becoming more interested in science. It's just the demystifying is what really matters, but also being excited about it and seeing the world around you through the lens of a scientist, asking questions, being eternally curious. I think that's really what it's much more about. But what about all the stuff about materials, Erum? I mean, that was the stuff that just blew my mind.
Yeah, of course, materials are super important. I love the fact that she's looking at using biology at the nanoscale. She talked about batteries and how we can look at energy production differently. And she's also taking the materials into the environment, into medicine. That's all amazing that she is bringing this to the table as a way to underwrite those policy recommendations.
Yeah, absolutely.
I am just floored in a good way that this is something that they are bringing forth bold ideas to Congress and that they're being taken seriously. She talked about some bills that are being passed. We didn't get to deep dive into those specific bills, and we should have Angela back on or someone else from the commission on. But we need people like Angela to be able to have these conversations with those in our government, because it's super important to have people that you trust that are from all different types of institutions. That's why I really love the fact that they have Alexander Titus on the commission as well. But on the materials side, on the policy side, what are you thinking, Karl? I mean, this is something that we've been talking about for a while.
The policy side or the material side?
Both. Let's do this.
Yeah. So I'll address the policy side first, because I think one of the things that we typically say to people is, how are you talking to your representatives? We need to demystify what scientists do. We need representatives to understand that science and science-based businesses are building the economy, and that we hire people, and that typically wages are really good for these jobs. So I think it's really important for people to understand that this is not pie in the sky. As important as research is, and it's incredibly important, it does create jobs. It creates new fields. All the things that we take for granted, like GPS and cell phones and whatever we live in, somehow came out of basic research. So I think anything that we can do, and we do it, I mean, we're doing it through this podcast, to get more people interested in these technologies and helping representatives and policymakers understand that these technologies are not to be feared. I think we're doing our job. In terms of the material side, look, I love what Angela's doing. I'm a huge fan of using biotechnology to build new things. And I just want to do a shout out to the book, The Age of Living Machines by Susan Hockfield. So the author, Susan Hockfield, is an American neuroscientist, but she was the president of MIT from 2004 to 2012.
Wow.
She wrote this fantastic book called The Age of Living Machines that really gives an overview of using biology as a building tool, as an engineering tool, in the way that only someone from MIT could do. So shout out to Susan Hockfield. Angela's story is also prominently mentioned in that book. So we'll leave a link to it in the show notes, and it's also worth reviewing.
Well, I mean, this circles back to the living machines and simulation. I'll just say that could mean a lot of things in today's age. At least that's what the algorithm's feeding me. But now this is, hey, look, whatever, if simulation or not, we're here. We're living in this world, and it's exciting, especially when we're meeting incredible people like Angela and the people that are on our podcast, and that we can build things. We could build things within the simulation, and hooray, good for us. But I think that was a great episode. Let's get some more people on that are in government, that are spearheading these bills, that are promoting the use of biotechnology and biomanufacturing, because that's how some of this is going to manifest. And it's going to be great to see who's working on the ground.
Yeah, and I would recommend that if you want to dig deeper into the policy or the conservation stuff, definitely go back and listen to the episode with Alexander Titus or the episode with Ben Novak of Revive and Restore. If you want to reach out and suggest guests or suggest topics, we do get a lot of people sending us suggestions. We really appreciate those.
Yes. And let us know if any events, we talk about going to different events or conferences and meeting people in person. We want to make sure that people know that we're actual people and we're not just AI chatting with you, which is fine. We have to respect AI too. Got to say that to protect my butt. But that's it. So thank you so much for listening. You have been listening to the Grow Everything podcast.